So the objective is to do even better, to further translate the biologically relevant information into even more effective and less toxic approaches. And to accomplish this, we'll molecularly profile the cell populations in the tumors to try and identify these progenitor or stem-like cells that are repopulating the tumor after successful treatment with shrinkage of the tumor, knowing that a certain percentage will come back. And the question is why? And it's because there are two possible explanations for that. That in the original tumor, there's a mixed population of cells, and that any of those populations can grow after the tumor's been knocked down. Or the alternative explanation is now evolving that appears to be the correct interpretation from a lot of data, is that, again, there's a mixed population, but there are certain parts of the population that can be susceptible to our therapies, and those cells cannot sustain growth of the tumor, but there's a subpopulation that is able to do that. And it turns out these are these progenitor or stem cells, and they are wired by nature to avoid toxins, and they're wired for a very important reason to avoid toxins, because they represent their close relative, where they came from to begin with, are cells that are meant to repopulate normal tissues once you've got injuries. Think about the organs that get in touch with the environment day in and day out: the skin, the intestinal tract, the lungs, the kidney. All of those cells turn over, and there's a population within each one of them that allows for that turnover. And if the genes that regulate growth get altered in those cells, it's still a stem cell. It still contains the ability to keep toxins out of it, but now it's a stem cell gone wild. And that's why our traditional approaches can shrink the tumor down for many patients, but then have it come back, because now that population will repopulate the tumor, and it's more resistant than the mixed population we had to begin with.